All right. Thank you, Russ, and thanks to Russ and Kathy and Natalia and the rest of the CERSI team for putting together what's been a really fantastic visit. We're very excited about the ability to work together going forward. I should also say that this is the first time I've ever seen somebody's goal being to create an FDA presence. Usually it's the opposite. So this is sort of new territory for us. All right, and it's important to realize that, what we're talking about here with the Precision Medicine Initiative and how we're going to approach next generation sequencing, what we've been talking about is our new policy and formation. So nothing here is finalized policy. So nothing I talk about in terms of forward-looking policy. This is not anything anybody has to do at this point. So I want to start with a few basics to talk about how we regulate in vitro diagnostics now and what our sort of legal and conceptual framework is for that. So this is the definition of in vitro diagnostics devices, taken from a very exciting book, the Code of Federal Regulations. So there is a legal definition of devices, and this is what we use. It's those reagents, instruments, and systems intended for use in the diagnosis of disease, blah, 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 including a determination of the state of health in order to cure, mitigate, treat, or prevent disease. So I highlight those because I want to point out that this is sort of a very broad definition. And in fact, when we're looking at these, what's an in vitro diagnostic and how we're going to evaluate it, we are looking at how the device will be used. And here it says in a therapeutic product trial, which we see a lot for companion diagnostics, but really in any sort of clinical setting. And this intended use is typically pretty specific. It's, what analyte are you trying to detect? What is the type of result? What specimens are you testing, and from what population as well as a number of other pieces of information, and what are you trying to do or diagnose with that information. So intended use is really central to how we look at everything, and it makes a big difference. Now, within the framework of this broad definition, there are many things that are actually IVDs. So some things are obvious. Complete test systems and kits and instruments are IVDs, reagents. So it's not just the whole system itself, but the components as well that make up that test are legally in vitro diagnostic devices and are regulated. Software can be an IVD, as well as just some algorithms and calibrators and controls I've listed here too. So these are examples. And whether or not they are IVDs, again, depends on the intended use of that particular device. So now let me, because obviously we're talking about next generation sequencing here, let me sort of apply what I've just talked about to a next generation sequencing test and tell you what our thinking is right now about what part of it is a device, what part of it is not, because there's been a lot of confusion about that. So this lists I've sort of binned all the steps of NGS into some major categories. So if you think about, there are some things that should be pretty obvious that would be the device, the chemistry, making the library, performing the sequencing, so the instrument, and a lot of the software analysis, getting the FASTQ file and going on to VCF, that would all be, that software would be a device. Interpretation has been tricky because it's a very sort of complicated process and it looks different in many places. But what we focused on in looking at interpretation is not what an expert would do, how an expert would look at any particular piece of evidence or make a decision about a patient, but really the software and algorithms that are used. So there are products that will do sort of an automated interpretation. Those would be more devicey. as well as algorithms. So even one thing we're talking about, and I'll talk about it more in the context of the Precision Medicine Initiative, are things like the ACMG guidelines, which don't say how to evaluate any piece of evidence, but provide a framework, a set of rules. And so that's an important discussion we're going to have with all of you and the rest of the community. Let me just go back and say that there are certain things like the lab report, knowledge bases that are used, pre-analytical variables that may or may not be a device. And I think that's going to depend a lot on the configuration of any particular test and the intended use of the test. And so they're important to be aware of, but it's not there's no hard and fast rule at this point. Things like manual interpretation, when you sit down and look at the evidence, you've done a PubMed search and an expert makes a decision, that's practice of medicine and we don't regulate that. And basically archiving the results, say in EHRs, I mean, that's a very important thing, but not something we're really focusing on as a device. Now, when we look at a device, we have and you may have heard this term, this risk-based approach. And so when we classify devices, we are looking primarily, but not completely, at risk. So we have 3 classifications. Class 1, which would be very common low-risk devices, or even some instruments that are just used as components. Most of those don't even need to come to the FDA. They just register and list with us, and they can be marketed. They have to comply with what are general controls. And for a scientific audience, this can be confusing because controls do not mean control experiments, but they basically mean some sort of set of requirements or criteria. So registering and listing is a general control. Reporting adverse events is a control. So those are all things. And complying with the quality system regulations, having good manufacturing practices in place is a control. Class 2 tend to be more complex and higher risk. Those do generally require applications to us in what's called a 510K. They need to be demonstrated that they're equivalent to an existing device in the market, substantially equivalent, so not identical, but just have the same properties in the end. Those are subject to what are called special controls. So that means that in addition to the general controls, there are special things you have to do in the case of a class 2 device. And so when we clear a class 2 device, we often will list out what those are. So they may be doing certain sorts of studies before you market or something like that. Class 3 devices are the most complex, and typically the highest risk. Those are like companion diagnostics for oncology are all class 3. Those require a pre-market application, and you have to demonstrate that those are safe and effective. So that's so you hear the word safe and effective a lot from us. So what does that mean? And those are defined again legally in our the Code of Federal Regulations. Safety being that there is reasonable assurance that the probable benefits outweigh probable risks, and effectiveness, essentially that you're producing clinically significant results, that there's meaning to the analytical result that's being produced. And again, what that actually looks like for any device is going to depend, again, on the intended use of that device. And an evaluation of that device and the evidence supporting its use. So when we look at a device, what are we looking at? Well, there are essentially 3 things. And again, this is all based, what it looks like exactly, it depends on intended use. There's analytical validity. So are you detecting the correct analyte? Are your results precise? Are they accurate? And I've listed some of the types of experiments information we typically require, so the types of validation studies. But this is not a complete list. And then clinical validity, where we're worried about predictive values as well as sensitivity and specificity. We want to ask, are you identifying the right medical condition or disease? Or in the case of actionability, are you predicting who responds and who doesn't? in the IVD world, we actually don't have a lot of sort of prospective randomized clinical trials. A lot of the evidence we see is from third parties, like from literature. Often, a lot of the studies are retrospective studies on baked specimens. But the type of clinical study, again, depends on what the intended use is and what evidence is available. So that is pretty flexible. And then labeling is also something we think about a lot, because at FDA we are, of course, concerned with the claims that people are making about what they are producing and selling. And so the claims that are made have to be truthful and accurate, and that includes the claims that are made about analytical and clinical validity. Those have to be supported by evidence. That evidence is often contained in the labeling and in our decision summaries for these devices, which are publicly available in FDA databases. So just before I move on to the precision medicine initiative, I just want to summarize by saying that, so FDA review does two things, one of which I think is pretty well accepted, that we're promoting and protecting public health by looking at safety and effectiveness. The other, and I think this will be an important discussion going forward, is that as we are moving into this world of precision medicine, where we're accumulating different evidence, I think having this sort of review of how IVDs perform is going to be very important for good science. We're all going to be looking, a lot of people are going to be looking at big data, getting data from different sources. Right now we see IVDs across the spectrum being used, but we don't know when, you go look at say, variants in a database, you don't know the analytical quality of those. You don't know what test was run, how the data was, how that variant call was generated, who generated it, or the analytical properties of the test that was used. And so I would submit to you all that as we think about learning from practice, we need to think about providing the base of data, a quality base of data. You would be very concerned if you designed an experiment in a lab where you were generating data that had some of the properties you might find in some public databases, for instance. So let's move now to talk about precision medicine. And I just wanted to start by contrasting sort of what I just talked about more or less, which really has grown up around traditional diagnostics, where you have essentially what I'm calling low to medium resolution technology. You're identifying a sort of a finite number of analytes, and that usually divides people into fairly large size subpopulations when you do those tests. Usually it's one test for one disease, and we're evaluating a single test at a time. And typically people do research, generate the evidence, and then market. Well, for precision medicine, we're really dealing with a new world. So we have high resolution technology. I mean, we have technology that can divide people into almost N of 1 cohorts in many cases. You have essentially an infinite number of variants you could be looking at. I mean, it's an undefined number of variants, and we can never predict all of them. For NGS, you run one test, who knows what you're going to diagnose? Many possible diseases. And a lot of the clinical evidence in the future is going to be obtained not from doing controlled studies, but from accumulating evidence from the community, from clinical practice. And that's really a new paradigm that we have to think about, for the future. So I don't really need to tell you about NGS, so people talk about precision medicine, and really we focused on NGS because that is really what at this point in time is driving precision medicine in the technology, certainly in the IVD world. But certainly what we're talking about, we anticipate we will have to apply in some form to the other omics, other high-resolution technologies and high-throughput technologies as they get developed and enter the clinic. So I think I've sort of alluded to some of these barriers to precision medicine. One is that the technology is still developing rapidly, and our understanding of the technology is still developing rapidly. So that means There are many different configurations of a test, all which may be perfectly legitimate. Tests are frequently modified, and the underlying technology is still evolving. And I think the end result of this is that we can see different labs, both of which can be really good labs, will still obtain different results from the same sample. And so this is a challenge. I don't think it's hard to know at this point who's right or who's not. And they even two very good labs will interpret the same variant differently. There's also, again, these unlimited results and this unprecedented ability to detect rare variants, which really makes it hard, if not completely unfeasible, to do clinical studies in many cases. Now, this poses issues for regulation. And I would say the big one is that there's no specific intended use in many cases we can wrap our heads around here. And that means, we have to think of new ways in which we're going to handle this, because a lot of our traditional requirements center around very specific intended uses, where we can evaluate each claim a test makes and each modification that is made to a test. We don't think that's a very efficient way moving forward, and not a way that's going to move things forward. Now, in the interest of time, because I want to get to what we're proposing and spend less time on history, but I did include some slides here on what some of the ways we've coped with some of these problems already, even in our traditional framework. We've certainly been able to evaluate next generation sequencing technology. We approved at the end of 2013, as many of you know, the MySeq DX platform from Illumina, as well as the chemistry. We cleared that. We also cleared 2 assays for cystic fibrosis, one which detects specific variants that are known to cause disease, and the other which is an assay that detects any variant in the entire cystic fibrosis, CFTR gene. Now, just to briefly summarize, we were able to look at in analytically validating the platform, we looked at a subpopulation of variants that represented different, easy and hard to sequence regions and different sort of types of variations to try to infer from representative performance that the whole platform was analytically valid. And we did that because, of course, we couldn't require analytical validation or evaluate it for every possible variant. Those can't be predicted. For targeted sequencing, we sort of used the same approach, but now focused on the cystic fibrosis gene itself. In the case of the variant assay, the specific clinically relevant variants. For clinical validity of the cystic fibrosis variant test, we used a public database, the CFTR2 database that was developed at Hopkins with support of the Cystic Fibrosis Foundation. And I've listed the types of evidence that contained. It was a really good database, and we ended up essentially reviewing the database rather than reviewing the clinical evidence. And I think that's an approach we want to expand on going forward. Again, just in the interest of time, I'm going to move on to the PMI, because I do want to talk about that, but I think it'll make sense now that there's some history and framework for that. So the PMI was announced at the beginning of this year in January, and it's a broad initiative. You've probably all heard of the 1 million person cohort that was recently announced. The vision of the PMI is really to do a number of things, to develop a number of things, but really the goals for us are to get the safe and accurate diagnostic tests, to help support the development of learning health systems that will enable evidence collection in the future, developing targeted therapies, because the whole point of this is to figure out how patients should be treated more effectively. And to do all this, we're focusing on what are our research and regulatory policies, but also research policies that will help bring about this new world. And so in terms of modernizing this, I think I've already gone over this. Our goal, again, is to make sure that patients and providers get accurate data that they understand. And so our approach in the near term is to really try to develop standards analytical standards and resources that will allow anybody to have a clear bar. Like, if I'm doing NGS sequencing, if I'm developing a test, this is sort of the minimum quality I need to get. So that's something a lot of people in this field are already working on. We want to leverage that and be able to come up with standards that will assure that in a regulatory sense, and then in the longer term, implement an actual system, a set of policies and processes that will actually allow laboratories and others developing these tests to comply without having to come to FDA for each and every claim for a test or each and every version of a test. So to sort of accommodate this broad use. And so I've mentioned some of these standards to ensure quality, open source tools. So we believe these will all lead to a more flexible and dynamic regulatory system, one which will also keep up with the the pace of knowledge generation, both technically and clinically. We also want to help support the development of high quality, publicly available databases, to enable the clinical interpretation. One of the big barriers here is also what people understand and the variety of things people are reporting. So I've provided more detail here, and I'm really sort of have completely miscalculated the time on this, so I apologize, but I'm happy to answer questions about it. So if I can just take a minute to wrap up. One of our big efforts right now is to develop technical standards for NGS around a specific intended use, and then to working with the scientific community and then expanding that to multiple intended uses. We are also for precision. We are also trying to develop open source tools, and this is what Precision FDA is about. We want, in the end, the goal of Precision FDA is to help developers meet those standards, and that's one of the things we really are excited about working with Stanford on is how would that actually look? What are useful tools? So that's our vision. We now have to fill in the details. And in a separate effort, we are beginning to work on what would a quality database look like. So there are curated databases like ClinGen or PharmGKB. For regulatory purposes, what would the quality standards look like? And then we have a number of ideas around that in terms of having trained personnel and proper standard operating procedures, but without getting down to the level of practice of medicine. So let me finally conclude by saying we've also announced on November 12th and 13th more workshops on analytical and clinical validity, respectively. Those can be seen on webcast if you can't be there in person. And there's going to be around some white papers we hope to release in a couple of weeks that will discuss more specific plans around what analytical and clinical standards for hereditary genetics will look like.